Hello everyone, myself Dr. Suresh, Professor of Biochemistry, and in today's video we'll be discussing about the metabolism of ammonia, or you can say detoxification of ammonia and the urea production. So in our previous videos we have already discussed how ammonia is produced via transamination and then deamination. Transamination is a mechanism how the produced ammonia will be transported from peripheral tissues to liver, and deamination, whatever the dumped amino acids in the form of ammonia, they will be deaminated to produce free ammonia, and this free ammonia will be diverted for urea production, right? So first we will see how the ammonia is produced and how this ammonia is trapping in the peripheral tissues and how it is transported to liver. And finally, once it reaches to the liver, how urea synthesis is taking place, right? So peripheral tissue, especially if you take from muscles, okay, one thing I'll talk about here, reduced production means whenever there is a production of high concentration of ammonia, so transamination reactions will come into the action. So they decrease the production or the quantity of ammonia in circulation, okay. And second thing is ammonia trapping. How this ammonia trapping is happening, that we'll see. So here, whenever there is a high concentrations of ammonia in the circulation, especially because ammonia is toxic to central nervous system, especially to the brain. So the major defense mechanism in detoxification of ammonia in brain via the formation of glutamine, okay. So the trapping of ammonia, especially in brain, via glutamine formation. What is the difference? What is this glutamine, and what is the difference between glutamic acid and glutamine? You see here, glutamic acid, it is having one amino group already, okay. And when there is excess of ammonia, glutamic acid is having a extra carboxylic group, so it can accommodate one more amino group because of this carboxylic group. So the extra amino group will be accommodated via carboxylic group of glutamic acid, right? So here you see the ammonia accepted by the carboxylic group of glutamic acid, and it is a energy dependent reaction, okay. The enzyme involved here is glutamine synthase, because we are producing what? Glutamine, right? So this glutamine were having two amino groups. You see here, one amino group is here and another amino group is here, okay. So what is happening here? Glutamic acid, original amino group it is having, and in glutamine it is carrying ammonia, okay. So the glutamine formation is mainly for the transportation of ammonia in non-toxic form from brain to liver, okay. So this way majority of the ammonia will be transported in non-toxic form via glutamine formation. So once it reaches to the liver, in liver cells there is enzyme glutaminase which acts on the glutamine to remove the added ammonia to form glutamic acid. So the removed ammonia will be channeled to urea formation, okay. So this way ammonia is trapping in brain. So there is one more system, like we have already seen in carbohydrate metabolism from peripheral tissues like muscles if you take, okay. There you can say glucose alanine cycle. Glucose alanine cycle. So what's happening here in skeletal muscle, first glucose converting into pyruvate. So if there is excess of ammonia, so this ammonia added by pyruvate to form alanine, okay. This is happening in skeletal muscles, okay. And this alanine allowed to transport in circulation and reaches to liver, okay. In liver, this alanine by deamination, okay, it can be removed ammonia and converted to pyruvate, and this pyruvate entering into gluconeogenesis and then converted to glucose, and this glucose again supplied to skeletal muscles, and removed ammonia will be involved in or channeled in production of urea. So there are two mechanisms for peripheral tissues, glucose alanine cycle, especially from skeletal muscles, how ammonia is transported via glucose alanine cycle. From the brain, it is glutamine formation, okay. The enzyme involved in making of glutamine is glutamine synthetase, which is energy dependent reaction, and once this glutamine reaches to the liver cells having an enzyme glutaminase which cleaves the added ammonia and forms the free glutamic acid. Right. Again, this glutamic acid further undergo cleavage by glutamate dehydrogenase, form one more ammonia. Okay, and this is also channeled to urea cycle. Why ammonia is toxic? So that we will see once we finish the urea cycle pathway. Okay, and now we will see what is the fate of this liberated ammonia in liver cells. Okay, so there is categories are there based on the excretory product. Okay, so amniotic, uricoteric, ureoteric. So amniotic means fishes, reptiles. You see, they don't have facility of urea cycle, so they directly excrete ammonia. Fishes and aquatic animals, they release directly ammonia. And uricoteric, like birds, okay, and reptiles, so they directly excrete uric acid. So their excreted product is uric acid. So that's why they are known as uricoteric. And we are excreting not ammonia but converting form of ammonia, that is urea. Okay, because ammonia is toxic, which is not allowed in our body, so it has to be converted into solubilized or non-toxic form urea. So that's why the chief content of our urine is urea. So we comes under category ureoteric. So there are three categories based on the excreted product: amniotic or ammonioteric, uricoteric and ureoteric. Okay, so now we will see the fate of liberated ammonia in liver cells. Okay, as we all discussed, liver is the detox center and how it detox the liberated toxic ammonia. Okay, so here ammonia combines with carbon dioxide to form carbamyl phosphate. First of all, we'll just go through few basic points about urea cycle. So urea cycle is the cycle discovered by the same scientist who has discovered TCA cycle, Sir Hans Krebs. Okay, so Sir Hans Krebs is the one behind urea cycle and Krebs cycle. Okay, and this is the first cycle to be elucidated by Sir Hans Krebs. Okay, later he has elucidated the TCA cycle. Okay, here the urea cycle, otherwise known as citrulline ornithine cycle. Okay, citrulline ornithine cycle. Okay, for exam point of view or competitive exam point of view, it is very important cycle. So what is the starting material for, as like TCA cycle, if you take the starting material is acetyl-CoA, which has to combine with oxaloacetate to form citric acid. So same way in urea cycle also there should be a starting substance which has to be combined with the existing substance. Okay, and any pathway before you study, okay, you have to note down the location of the pathway and the cells. So we already know which organs this urea cycle is taking place: liver cells, especially liver cells, this urea cycle is taking place. Okay, and the location inside the cell, where? So we have mitochondria and we have cytoplasm. So where these pathways take place? So if you talk about glycolysis, glycolysis is a cytoplasmic pathway, and if you talk about TCA cycle, it is a mitochondrial pathway. But here urea cycle is partly mitochondrial and partly cytoplasmic. So here first two reactions, first two reactions are taking place in mitochondria, rest of the three reactions are taking place in cytoplasm. Okay, so two reactions in mitochondria, last three reactions in cytoplasm. So first we'll see one by one. Okay, the steps of urea cycles. For ammonia, first combine with carbon dioxide to form carbamyl phosphate. So amino group, nitrogen is there, ammonia is carrying nitrogen. This nitrogen combines with carbon dioxide, okay, by the enzyme carbamyl phosphate synthetase 1. Okay, it has to be carbamyl phosphate synthetase 1. Why I am saying carbamyl phosphate synthetase 1? Because there do existence of carbamyl phosphate synthetase 2. Okay, I'll tell the difference between carbamyl phosphate synthase 1 and carbamyl phosphate synthase 2 here. If you see the location, okay, CPS1 and CPS2 both differ in their location. Okay, CPS1, as I told you, urea cycle first two reactions are taking place in mitochondria. So obviously CPS1 is located in mitochondria and CPS2 is located in cytosol. And now where these enzymes involved? So CPS1 obviously in urea cycle and CPS2 involved in pyrimidine synthesis. Okay, CPS2 involved in pyrimidine synthesis, and there is a positive effector for CPS1, the enzyme, that is N-acetyl-glutamine. Okay, but in case of CPS2 there is no such facility. There is no positive effector for CPS2, and sources for nitrogen of CPS1 is ammonia, but in case of CPS2 the source of nitrogen is glutamine. And the inhibitor, there is no such inhibitor for CPS1, but in case of CPS2, because it is involved in pyrimidine synthesis, so there should be some control. So if there is excess of CTP, which has inhibitory effect on CPS2, so that pyrimidine synthesis will be regulated. Okay, so this way both the enzymes differ in location, pathway, and inhibitor wise. Okay, and the source of nitrogen value. So the first step, as I am talking, ammonia, okay, and carbon dioxide combine to form carbamyl phosphate, because here, okay, here there is involvement of ATP. Okay, here ATP is involved. Okay, so that the phosphate group is donated to here, so ATP is converted to ADP. Okay, so and carbamyl phosphate synthetase, okay, and ammonia, carbon dioxide to form carbamyl phosphate by the enzyme carbamyl phosphate synthetase. And next step, the ornithine, which is the regenerative substance of urea cycle, involved in the cycle. So because in urea cycle, oxalo acetate in the first step will be involved, but in here urea cycle, in the second step ornithine, because in the TCA cycle, oxalo acetate is the first to be involved and last to be regenerated. Here ornithine is involved in second reaction and the last it will be regenerated. Okay. So by the enzyme ornithine transcarbamylase, this enzyme is also present in mitochondria. When carbamyl phosphate combines with ornithine, it forms citrulline, and there are citrulline transporters available in mitochondria, so that citrulline, whatever formed in mitochondria, can be shifted to cytoplasm. Okay, and once this citrulline is transported out of mitochondria, aspartate, the amino acid aspartate added to the citrulline by the enzyme 𝐒𝐮𝐻𝐼NO synthetase. So this is the cytoplasmic enzyme. So as I mentioned, two enzymes, first two enzymes, carbamyl phosphate synthetase 1 and ornithine transcarbamylase, both are mitochondrial enzymes, and rest of the reactions which are taking place in cytoplasm, so those enzymes belongs to cytoplasm. Okay, so 𝐒𝐻𝐼NO synthase, whole molecule of aspartic acid will be added to citrulline to making of 𝐒𝐻𝐼NO. Okay, and it's such a huge molecule, it has to be cleaved by the enzyme 𝐒𝐻𝐼NO lyase. So the succinate part will be converted into fumarate part. Okay, so note down this point, fumarate. Okay, and rest of the part will be converted into arginine. So we know arginine. Arginine is a semi-essential amino acid. It is a basic amino acid type. Okay, and this arginine acted by the enzyme arginase. Okay, so to form urea, and rest of the part will be converted to ornithine, which will be regenerated. See, so these three steps are cytoplasmic steps. So one enzyme, step 1, step 2 are mitochondrial, step 3, step 4, step 5 are cytoplasmic. Okay, this way urea cycle is taking place. Whatever the ammonia is liberated in the liver cells. So when you see in detail of the one by one, so as I mentioned, you see here ammonia, carbon dioxide, water molecule involved, and here the involvement of carbamyl phosphate synthetase 1, 2 molecules of ATP involved here. Okay, in making of 2 ADP plus PI, actually 2 PI has to be there. So 1 PI is already added to carbamyl group to form carbamyl phosphate. So obviously 2 ATP is converting into 1 ATP is converting into ADP plus inorganic phosphate. Right, same way one more ATP is also converting into ADP plus PI. So how many are there? 2 ADPs. Okay, we got 2 ADPs, but 2 PIs are supposed to be there, but only 1 PI is there. So that PI is added to carbamyl group, so to form carbamyl phosphate. So now it is equal to 2 ATPs. Okay, it is equal to 2 ATPs. So second step again, ornithine. You see, remember here ornithine. Remember this amino groups. So citrulline, this is the structure of, there is addition of this carbamyl phosphate to ornithine. So this carbamyl phosphate group has been removed. So this carbamyl group has added to the ornithine to form citrulline. Now this citrulline, again this incorporation of whole molecule of aspartic acid with the help of ATP. Here you see AMP and PPI, and one phosphodiester linkage breakdown is equal to 1 ATP. So here 2 phosphodiester linkages are, so actually ATP is a combination of AMP, PI plus PI. So adenosine monophosphate, if you add one phosphate, it will become ADP. If you add one more phosphate, it is triphosphate. Adenosine triphosphate. So one breaking off, when you convert ATP to ADP, that is equal to one, one phosphodiester linkage, which is equal to ATP. Same way here, you are breaking two phosphodiester linkage, which is equivalent to 2 ATP. So initially we have spent 2 ATPs, and here though it is a single ATP, but we are cleaving two high energy phosphodiester linkage, so which is equal to 2 ATP. So total so far 4 ATPs we have invested. So you see here, whole molecule of aspartic acid is added to citrulline. This is citrulline, and whole molecule of aspartic acid added by the enzyme 𝐒𝐮𝐻𝐼𝐴 synthase. Here 𝐒𝐻𝐼𝐴 lyase, so what is happening here, it is 𝐒𝐻𝐼𝐴. So this is a succinate group. So this part has been removed as fumarate. So as I told you, remember this part fumarate. What is the fate of this fumarate? This fumarate can be converted to malate, and again malate can be converted to oxaloacetate, and oxaloacetate can be converted to aspartic acid, okay, and again rejoins the urea cycle, otherwise it can go into the TCA cycle. So there are two fates of this fumarate based on the cellular demand. Okay, now the molecule of arginine is there. Okay, so this is a guanidino amino group with the help of the arginase. So this part NH2 C double bond O NH2 with the help of addition of water molecule, okay, there is again regeneration of ornithine, there is a formation of urea. Okay, so simple. Don't pay attention to the molecular chemical structures, so until unless you are clear, otherwise with the name you can remember. So now what I am talking about here, as I told you, fumarate can be entering into conversion to by the enzyme fumarase to form malate, and this malate can be converted into oxaloacetate by malate dehydrogenase, and again it can convert to aspartate. Again aspartate again will be involved in TCA. So that is a link between TCA cycle and urea cycle. So both are together. Okay, TCA cycle and urea cycle will go together. Okay, so you can compare this as a bicycle. Front wheel as a TCA cycle, back wheel as a Krebs cycle. So to summarize here, whole pathway, urea cycle, which is on the there are in the colored one, two reactions in mitochondrial, and green color which is cytoplasmic, and again the pink color what I am showing the link to TCA cycle and urea cycle. So to remember 5 enzymes: carbamyl phosphate synthase 1, ornithine transcarbamylase, 𝐒𝐻𝐼𝐴 synthase where aspartic acid added, and 𝐒𝐻𝐼𝐴 lyase, next arginase. So 5 enzymes you have to remember for urea cycle. So to summarize overall reaction, NH3 plus CO2 plus aspartate gives urea and fumarate. In the urea cycle, 2 ATPs are used in first reaction. Another ATP is converted to AMP and PP, which is equal to 2 ATPs. So net thing is we are spending 4 ATPs. Okay, so one urea cycle consumes 4 high energy bonds. However, fumarate formed in 4th step may be converted to malate, and malate when oxidized to oxaloacetate produces 1ADH equal to 2.5 ATPs. So 4 ATPs minus 2.5 ATPs, that means our net invest is 1.5 ATPs we have invested from our pocket. 2.5 ATPs has been recovered via malate to oxaloacetate formation. The urea cycle and TCA cycle are interlinked, so it is called urea bicycle. So course regulation, how this urea cycle will be regulated. So during starvation, what's happened, a lot of energy is required, so most of the proteins will be degraded and amino acids will be catabolized, and carbon skeleton will be involved in gluconeogenesis, and ammonia will be produced, right? So to meet the increased energy demand, right? So urea cycle enzymes elevated in case of starvation. Okay, urea cycle, because most of the proteins are catabolizing, amino acids are catabolizing, so obviously there is a high production of ammonia, so it has to be converted into urea. So in starvation, urea cycle enzymes are elevated. And fine regulation, like a majority regulator enzyme is CPS1. It is a rate limiting step or the committed step of the urea cycle. So and the activator is N-acetyl glutamate, will stimulate the reaction. It is formed from glutamate and acetyl-CoA. And arginine is the activator of NAG synthase. So N-acetyl glutamate is activator. So arginine when you give, it stimulates NAG synthase. So when NAG is there, CPS1 obviously will be active and further urea cycle will be proceeded. Compartmentalization. So this way also there will be regulation over urea cycle. Urea cycle enzymes are located in such a way that first two enzymes in mitochondrial matrix and rest of the three in cytoplasm. So the inhibitory effect of fumarate on its own formation is minimized because 𝐒𝐮𝐬𝐜𝐫𝐞 lies in the cytoplasm while fumarase is in the mitochondria. So it may not be having much efficiency inhibitory effect because 𝐒𝐮𝐬𝐜𝐫𝐞 lies is in cytoplasm and fumarase is in mitochondria. Deficiency of any of the urea cycle enzymes would result in hyperammonemia. So when there is a block in one of the earlier steps, the condition is more severe since ammonia itself accumulates. So it's called hyperammonemia. So as I told you to remember 5 steps, so one step defect causes one type of hyperammonemia. So urea cycle disorders are 5. So 5 steps, 5 urea cycle disorders: hyperammonemia 1, hyperammonemia 2, citrullinemia, 𝗠𝐼𝐻 aciduria, 𝗠𝐺𝐼nemia, and ornithinuria, hyperarginemia, 𝗠𝐺𝐼nic aciduria. So deficiency of later enzymes results in accumulation of other intermediates which are less toxic and hence symptoms are less. As a general description, disorders of urea cycle are characterized by hyperammonemia, encephalopathy and respiratory alkalosis. Clinical symptoms including vomiting, irritability, lethargy and severe mental retardation. Infants appear normal at birth, but within days progressive lethargy sets in as ammonia starts working on central nervous system. Treatment is mainly symptomatic. Low protein diet with sufficient arginine and energy by frequent feeding can minimize brain damage since ammonia levels do not increase very high. Why? Because arginine involved in making of NAG, stimulates NAG synthase, and when NAG synthase is activated, more NAG formation, and this NAG, N-acetyl glutamate, stimulates CPS1 to convert ammonia into next product, that is carbamyl phosphate. So ornithine transporter deficiency is characterized by hyperornithinemia, hyperammonemia and homocitrullinuria. Since ornithine is not available in the mitochondria, lysine is carbamylated to form homocitrulline. Why brain is very sensitive to ammonia? So here in children, if you see particularly, may put on a low protein diet and a frequent small feeds has to be given. Attempt may be made to eliminate the amino, nitrogen and other forms like hippuric acid, like giving benzoyl conjugate of glycine or phenylacetyl glutamine. So this way you can reduce the concentration of ammonia to minimize the effects of ammonia on central nervous system. Since citrulline is present in significant quantities in milk, breast milk is to be avoided in citrullinemia. Ornithine transcarbamylase deficiency is the only urea cycle disorder which is inherited as X-linked. So inborn error, that means which is ornithine transcarbamylase, because this is a mitochondrial enzyme, and second step of urea cycle which is X-linked trait, which is inherent in nature. So elevated levels of ammonia are associated with high glutamine levels in cerebrospinal fluid and blood. 𝗠𝐺𝐼nosuccinate lyase deficiency leads to 𝗠𝐺𝐼nosuccinic acidemia and therefore metabolic acidosis it is causing. Hyperammonemia is less severe and 𝗠𝐺𝐼nosuccinate is elevated in central CSF and excreted in the urine, cerebrospinal fluid, and which has to be excreted in the urine. In case of arginase deficiency, it is a most mild variety with accumulation and excretion of arginine which has to be seen. Symptoms appear by 2 to 4 years of age. The accumulation of ammonia in blood, normally less than 50 micrograms per dl, body fluids results in toxic symptoms. Nowadays, defects in enzymes of urea cycle are detected in neonatal blood by estimating metabolites by tandem mass spectrophotometry, or you can say tandem mass spectrometry. So when you see the tabular column, compare the urea cycle disorders. Like as I told you, 5 steps are there, 5 disorders are there. Urea cycle: hyperammonemia type 1, hyperammonemia type 2, hyperornithinemia. Next is citrullinemia, 𝗠𝐺𝐼nosuccinic aciduria, and less variant, that is hyperarginemia. Okay, so first two, hyperammonemia type 1 and hyperammonemia type 2, are severe, okay, and they are lethargic. So the enzyme deficiency in hyperammonemia is first reaction, that is CPS1. So in this, ammonia itself accumulated, so high levels of ammonia you can see in blood. It is autosomal recessive, mental retardation, incidence 1 in 1 lakh. And hyperammonemia 2, ornithine transcarbamylase deficiency, where carbamyl phosphate cannot be incorporated into ornithine. And here also ammonia accumulation is high, in blood and increase in glutamine concentration in blood, CSF and urine, orotic aciduria due to channeling of carbamyl phosphate into pyrimidine synthesis, and also it is an X-linked one. So this is X-linked ornithine transcarbamylase, which is seen in hyperammonemia type 2, okay, which is X-linked. We can see in only in males. Okay, and orotic aciduria, because carbamyl phosphate is not adding to ornithine, so the accumulated carbamyl phosphate channel to pyrimidine synthesis, and it can be causes more amount of orotic acid, and which can be seen in the urine. On hyperammonemia, hyperornithinemia, and here because whatever ornithine from cytoplasm to again get back to mitochondria, because ornithine is producing in the regenerating in the cytoplasm, and whatever ornithine is regenerated in the cytoplasm has to come back to the mitochondria. So here if there is a defect in the transporter of ornithine from cytoplasm mitochondria, what happened if there is no ornithine, what happens again, carbamyl phosphate will be high in nature, and it is also orotic aciduria, again one of the common symptom, common future. Failure to import ornithine from cytoplasm mitochondria, defect in ornithine gene, hyperornithinemia, hyperammonemia and homocystinuria is seen, decreased urea in blood, autosomal recessive condition it is. So next one is citrullinemia. So citrullinemia is because of deficiency of the enzyme 𝐒𝐼𝐻𝐼𝐻 synthase. Because of the 𝐒𝐼𝐻𝐼𝐻 synthase deficiency, aspartic acid cannot be added to citrulline, so whole molecule of citrulline will be accumulated, and you can see excess amounts of citrulline in the blood causing citrullinemia. It is also autosomal recessive inheritance, which has high blood levels of ammonia and citrulline. You can see citrullinuria 1 to 2 grams per day normally. Okay. And 𝗠𝐼𝐻𝐼𝐻 aciduria, because of the enzyme deficiency 𝗠𝐼𝐻𝐼𝐻 lyase, and you can see high levels of 𝗠𝐼𝐻𝐼𝐻 in blood and urine, and friable brittle tufted hair, trichorhexis nodosa. Incidence is 3 out of 2 lakhs. Hyperarginemia, again arginase, arginine increased in blood and CSF. Instead of arginine, cysteine and lysine are lost in urine. Incidence 1 in 1 lakh. Okay, along with arginine, cysteine, lysine is also lost in the urine. So hyperarginemia, out of 5, one is a transporter defect, otherwise rest of the disorders are defect of enzymes of urea cycle. Okay, so that's all about urea cycle. So thanks for watching. Thank you.